Hello mga ka-Block D! Welcome to my YouTube channel. Thank you for watching it. Charot. So today, we are going to discuss about the experiment 5 and 6, which comprises the qualitative test for carbohydrates and the identification of the unknown. The objectives for this experiment will be flashed on the screen. Just look at it, read it. So carbohydrates have the general formula of CnH2ON, where N is equals to 3 or more. However, this formula is not applicable for all carbohydrates. It's for monosaccharides only. So they are considered to be the main source of energy in the body, and cells that utilizes carbohydrates as main source of energy in the body are brain cells and red blood cells. So most carbohydrates occur as polysaccharides. We have structural components, chitin, which are found in the exoskeleton of the insects, cellulose, which are found in plants. We also have carbohydrate storage polymers, which are starch and glycogen that are found in plants and animals respectively. Carbohydrates can be a polyhydroxyaldehyde, an aldose sugar, which comprises of one R group, one carbonyl group, and one hydrogen group, which make it a chiral carbon. Polyhydroxyketone, a keto sugar, has two R groups and one carbonyl group, and it is not a chiral carbon. So, three classifications of sugars. We have monosaccharides, the simplest sugar, oligosaccharides, and the polysaccharides. So, monosaccharides are the building blocks of carbohydrates. So, the simplest monosaccharides contain 3 carbon atoms, which are called as trioses. The simplest aldose is called as the glyceraldehyde, and the simplest ketose is called as the dihydroxyacetone, which can be seen on the screen, their structures. You can see a glyceraldehyde are composed of a carbonyl group, one R group, an OH group, hydroxyl, and we have an one hydrogen atom attached into it. While a keto group is attached to 2 R groups. And for oligosaccharides, so these means few sugar units. So we have disaccharides, trisaccharides, so on and so forth. And for polysaccharides, contains more than 10 monosaccharide units. It can be a homopolysaccharide or a heteropolysaccharide. So homo means it contains one type of sugar. For hetero, we have it contains more than one type of sugar. So carbohydrates, they undergo an intramolecular interaction, and the groups that are involved in that interaction are the hydroxyl group and carbonyl group. So for aldoses, it forms a hemiacetal, and for ketoses, it forms a hemiketal. So it's important to note that for aldoses, the carbon 1 and the carbon 5 are the ones who are involved in the interaction, whereas for ketoses, we have carbon 2 and carbon 5 that interacts. And there are 2 different forms of cyclic sugars. We have alpha and the beta. So we know naman that alpha, the OH group, the oxygen, the OH would be below. So you can see on the picture. And for a beta, we have the hydroxyl group, which is pointing upward. So you can see on the picture too. So it is believed that cyclic anomers are relatively stable than open chain, thus the importance of these interactions. Now let's go to the experiment. But before that, we should know these general classes of carbohydrate test reagents. We have 3: dehydrating acids, condensation reagents, and solutions which contains copper 2 ions. So dehydrating acids causes the dehydration of carbohydrates and leads to the formation of furfural derivatives, namely furfural, which forms from a pentose, and a hydroxyl methyl furfural from a hexose. So for fural derivatives, it means dehydration or removal of three molecules of water. So next are the condensation reagents. It detects the presence of the furfural derivatives from the previous process or the dehydration. So these are phenolic compounds that react with furfural derivatives that yields highly colored products. It includes Molish reagent, Bial arsenose reagent, Selewanov's reagents. Lastly, we have solutions containing copper 2 ions, which as we have seen in the lab, they are usually in the color blue. So these reagents are the ones that oxidize the certain carbohydrates in which copper 2 to copper 1. So hence these sugars are called as reducing sugars, which contains free aldehyde group or acyclic hemiacetal. So this includes Benedict's reagent, Barfoy's reagents, and Felling's reagents. So first test that was performed was the solubility test. So a pinch of a solid carbohydrate sample was placed or added with 2 ml of water and 2 ml of ethanol to see its solubility. So the solubility of carbohydrates is dependent on its polarities. A fructose is a keto sugar, a monosaccharide. A glucose is a monosaccharide sugar, which is an aldose, and a sucrose is a disaccharide sugar that is composed of alpha 1 2 glycosidic linkage of the glucose. So these carbohydrates, they are polar molecules. They have the hydroxyl group, making it attractive to the very polar water, dissolving the substance in the process. So their structure is also a lot simpler compared to the starch. So they have 6 to 12 carbon atoms only, making them highly soluble in water. So the OH groups of the carbohydrates are responsible for the solubility of the compound. So these sugars are only partially soluble in ethyl alcohol because alcohol is 30% water and 70% alcohol. So the ethyl alcohol only has one polar area and has a larger non-polar area. So the water and alcohol interacts with each other, which causes the water not dissolving with the sugar as it normally would than in pure like solutions. So starch insoluble in water and in ethyl alcohol because it is a complex polysaccharide made out of linear amylose and branch amylopectin, which monomer is glucose, D-glucose. So amylose chains form into double helices and is insoluble in cold water. So the branched form of amylopectin allows it to be soluble in cold water. So ang native starch is in the form of granules that is generally insoluble in cold water but is soluble in hot water. So its solubility in water can be increased through heat. So this can be called as gelatinization. So the granules swell and the fragment under heat treatment. So insolubility in ethyl alcohol is explained by water having more affinity to the alcohol than the starch. So this lowers the solubility and the potential hydrogen bonds. So Molish test, 5 drops of sample were added with 3 drops of the Molish reagent and 15 drops of sulfuric acid. A Molish test, so it is a chemical test used to detect the presence of carbohydrates in the analyte. So the positive result is the formation of the purple or purplish red ring at the interface of 2 layers, as can be seen in the picture. So Molish reagent comprises of alpha-naphtol in alcohol, so it causes the solution to yield a highly colored product. So it is a condensation reagent, as what I have said in the previous slides. And sulfuric acid acts as a dehydrating acids, which causes the dehydration of all carbohydrates in the analyte. So application of this test is that An improved alpha-naphthol test detects presence of ketosis in normal and diabetic blood, in normal urine, and in normal and pathological spinal fluids. From the experiment, these different samples that were tested and the following observations and the results. So as we can see, of course, water does not contain any carbohydrates at all, and the other three samples, well, of course, they are carbohydrates. Hence, they are expected to be positive, and it is reflected on the results. So, all carbohydrates will give a positive results. However, monosaccharides gives a rapid positive result relative to disaccharides and polysaccharides. So in general, monosaccharides are expected to react rapidly relative to disaccharides and polysaccharides. Thus, in the experiment, it was observed that glucose reacts faster since it is a monosaccharide. As for the sucrose and starch, it reacts slower than glucose. So why? Because unlike in monosaccharides, both disaccharides and polysaccharides needs to undergo hydrolyzation of its glycosidic linkages in the sugar molecules to monosaccharides. This is the mechanism of the Molish test. An example is the D-glucose being hydrolyzed first with the use of the sulfuric acid into hydroxymethylfurfuryl, and that furfuryl derivative was subjected to a condensation reagent, which in this case is the alpha-naphthol, and hence through the process, it gives a purple colored dye. So next is an iodine test. So a drop of dilute solution of iodine in Ki to 5 ml dilute sample were tested, and it was heated to boiling water. Then it was then cooled. Iodine test is an experimental test for polysaccharides, especially for starch. So a positive result is shown in the formation of the dark blue complex, and starch forms a blue colored complex with the iodine that was added. So when heated, the color disappears. So why is this happening? Heat causes the iodine starch complex to become unstable. The helical structure of the starch would uncoil and becomes linear. Hence, the iodine trapped inside the structure will be released and causes the change in color. So you can see the picture shows the starch iodine complex. So what can we infer from this? So the helical structure of the starch is significant for the formation of the complex. Thus, linearization of the structure disrupts the complex. The iodine molecules goes inside the coil that causes the formation of the colored complex. So heat causes the iodine starch complex to be unstable. Starch break down when heated to boiling point, and chains of the amyloses breaks. Thus, formation of short chains of dextrins causes the color to change. Heat causes the unwinding of the helical structure of starch, releasing the trapped iodine that causes the color of the solution to change. If the solution is cooled, starch structure becomes helical once more, and the dark blue complex is then restored. So in the case of amylopectin and glycogen, when it reacts with iodine, it does not yield a blue-black complex. It yields a red-brown complex instead. So monosaccharides and disaccharides would have a negative result because it is too small to be helically coiled. So application of this iodine test is for the hyperhidrosis, which is a common problem for amputees. It is also known as polyhidrosis or sodorrhea, in which a condition characterized by excessive sweating. So iodine starch test is commonly used to guide treatment decisions for hyperhidrosis. So this, as you can see in the picture, the one on the left part is the positive test. So when it was heated to boiling water, you can see that the dark blue complex starts to change until it becomes this yellow, slightly pale yellow liquid. But once it is being cooled, it starts again to change its color, then fully goes back to its dark blue complex. So next we have the fermentation test. So 5 drops of sample were subjected to 20% of suspension of ordinary yeast, then it was left for 30 minutes. So fermentation would result into the production of ethanol and carbon dioxide. So formation of bubbles indicates the presence of carbon dioxide. So yeast in general ferments the sugar glucose. So it specifically ferments the sugar. So as you can see from the results, arabinose, no bubbles formed. Sucrose and glucose, there were bubble formation, and galactose, there were no bubbles formed. So arabinose is a non-fermentable sugar. So, and it does not contain glucose too. It is a non-fermentable sugar. Okay. Arabinose is a non-fermentable sugar. For sucrose, it is a disaccharide composed of glucose and fructose. Hence, bubbles form due to the glucose unit present on that sugar. However, fermentation does not occur directly for the sugar sucrose. It needs to be broken down into fructose and glucose with the enzyme invertase that is present in yeast. Then, fermentation could be possible now with the enzyme zymase and ferments the sugar. So, glucose is fermentable, of course. It's the sugar that the yeast ferments. So, it was observed that there were more bubbles formed relative to the sucrose sugar. So, yeah, the enzyme zymase is the one responsible for making this fermentation occur. Maltose, which is a disaccharide, it's considered to be the best yeast metabolism because it contains 2 glucose units. Yeah. And galactose, on the other hand, no bubbles were formed. Yeast does not have the enzyme that breaks it. So the galactose metabolism, it needs the galc, galt, and gal. The galc, which is the galactokinase, galt, the galactose 1 phosphatidyltransferase transferase, and the UDP galactose epimerase. So it needs 3 enzymes for it to be broken down. So this is the reaction for the fermentation test. Glucose, yeast, it produces into ethanol and carbon dioxide that is shown in bubbles. Next test we have is the Seliwanov test. So 4 drops of the reagent, 5 drops of the sample were placed in a test tube. Then it was dipped in the boiling water at the same time. Then the changes were observed within 15 minutes of boiling, and the color were noted as well as its time of appearance. We have the Seliwanov test, a chemical test for aldoses and ketoses. So the positive result for ketoses, it reacts within 2 minutes and it forms a cherry red product. For aldoses, it reacts after 2 minutes, and formation of blue-green product also were seen, and also it is possible for it to change in peach color. So reagents composed of resorcinol and hydrochloric acid. Hydrochloric acid, again, a dehydrating acid. Resorcinol, a condensation reagent. It reacts with 4 phenol derivatives and thus formation of the highly colored product. So when heated, ketones are expected to dry out quickly than aldoses. So applications of this test is for glycosuria. It happens when you pass blood sugar, the blood glucose, into your urine. Normally, your kidneys absorb blood sugar back into your blood vessels from any liquid that passes through them. With glycosuria, your kidneys may not take enough blood sugar out of your urine before it passes out of your body. So this often happens because you have an abnormally high level of glucose in your blood, which is called as a hypoglycemia. Sometimes glycosuria can develop even if you have normal or blood sugar levels. So in this situation, it's known as renal glycosuria. So glycosuria, it normally occurs in pregnancy. So glycosuria is common in pregnancy. It's because of the lowering of the renal threshold of glucose excretion. Means very little or no sugar in urine during pregnancy. So here are the results from the experiment that we did. So as you can see, blank water, of course, negative. For arabinose, it is an aldopentose. A glucose is an aldohexose. Fructose, formation of dark orange complex, it is a ketohexose, hence it's positive. And sucrose, formation of light orange complex, a disaccharide, which comprises of glucose and fructose, hence positive test. Starch, no reaction or clear solution. So it is a polysaccharide. Of course, it would not yield a positive test. So ketosis normally gives you a cherry red color. So for aldoses, it gives the test weakly and slowly. So if the boiling is prolonged, positive test is obtained with glucose due to its partial conversion to fructose. So this test is also given by sucrose, which is hydrolyzed during the course of test, yielding fructose as one of the products. So hence the positive test of the sucrose. So why does ketose react faster in Selewanov's test? So ketose, when heated, they are dehydrated faster, hence they give the positive test faster relative to aldose sugars. So here is the mechanism of the Selewanov's reaction. It is shown on the screen. A ketose sugar, dehydrating acid was added, hence dehydration occurs, forming a porphyryl derivative, then added with the reagent, the condensation reagent, which is a Lewanov's reagent, hence the formation of the colored complex. This is the bile orsinols test. So we pipette a 0.5 ml of sample sugar solution, added with 4.5 ml of bile orsinols reagent, placed in boiling water, and the change in color were noted in 15 minutes of heating. So it is a chemical test for distinguishing pentoses and hexoses. However, this test is mainly sensitive for pentoses. So pentoses forms a blue or green complex, while hexoses forms a muddy brown to gray complex. So a positive result is indicated by the formation of blue-green complex. For negative result, we have the formation of any other color. So as you can see on the picture on the screen, the blue-green complex is the positive, then the other colors are not considered to be positive anymore. So the reagents is composed of orsinol and concentrated hydrochloric acid. So again, hydrochloric acid acts as dehydrating acid. It forms a porphyryl derivative, hence addition of condensation reagent, which in this case is the orsinol. It reacts with it and forms a highly colored product. So heating with strong acid converts the pentose to porphyryl, which then reacts with the colored compound produced when orsinol and ferric chloride react with each other. So a blue-green compound is formed. So this reagent reacts with many sugars, but under the condition described, only pentoses yield blue-green colors. So application is the blood galactose test. So the galactose 1-phosphate uridyl transferase blood test. So the galactose 1-phosphate uridyl transferase is a blood test that measures the level of a substance called GALT, which is an enzyme for galactose, which helps in the breakdown of milk sugars in your body. So a low level of this substance causes a condition called galactosemia. Here are the results from the bile orsinols test in the lab. Ching, 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 ching. So as we can see, the one that yielded positive test was the arabinose sugar. Yeah, of course. And for glucose, fructose, sucrose, and starch, we have negative. So why? Arabinose is the aldopentose sugar, and this test is specific for pentoses. So glucose is an aldohexose sugar, fructose is a ketohexose sugar, sucrose is a disaccharide, and starch a polysaccharide. Hence the negative test. So next, you can see on the screen the mechanism of the bile orsinose reaction. So a ribose sugar added with hydrochloric acids, then it gives off water. Dehydration of three water molecules. And then it forms a 4-furyl derivative addition of a condensation reagent, which is the orsinose reagent. Hence the formation of a blue-green condensation product. Positive test is the Felling's test. So 5 drops of the sugar sample were added with 5 drops of Felling's A, 5 drops of Felling's B, and then immersed in boiling water for 15 minutes. What is this test? It is a test for reducing sugars, an indicator for an aldehyde. So positive result is the formation of red precipitate. So that is the red precipitate. Copper oxide. Yeah. And as shown on the picture on the screen, the red one, that's the positive result. So reagents contains copper 2 plus ions in aqueous basic solution. So Felling's A solution is aqueous copper sulfate. Felling's solution B is an alkaline sodium potassium tartarate, or called as the Rochelle salt. So application can be used to screen for glucose in urine, thus detecting diabetes. So ketones cannot be oxidized using the Felling's reagent. However, Felling's test also reacts to ketones since they exist in chemical equilibrium with their corresponding aldoses. So in aqueous solution due to ketoenol tautomerism. So carbohydrates with free aldehyde or ketone groups reduce copper sulfate to cuprous oxide, forming a yellow or brownish red colored precipitates. So Felling's test can be used as a generic test for monosaccharides. So it will give a positive result for aldose monosaccharides due to the oxidizable aldehyde group, but also for ketose monosaccharides as they are converted to aldoses by the base reagent and then give a positive result. So for this reason, Felling's test can be considered as a general test for monosaccharides. So again, here are the results for the Felling's test. Water, blank, negative. Glucose, positive, since glucose is considered as a reducing sugar. So galactose yields a positive result too because it's a reducing sugar. Sucrose, negative, because it is a disaccharide. Lactose, positive, since lactose is a reducing sugar. Although it's a disaccharide, it is a reducing sugar because it contains a glucose unit which is free to interact with the oxidizing agents. Lastly, starch, of course, negative result. It is a polysaccharide, guys. Mechanism of the Felling's test. We see the ditartarate complex reacts with an aldehyde, hence formation of a carboxylic acid and the red precipitate, the copper oxide. So next test performed was the Barfoed test. So Barfoed test, it is a chemical test for distinguishing reducing monosaccharides from reducing disaccharides. So monosaccharides forms copper 1 oxide, which is the red precipitate within 2 minutes, and for disaccharides more than approximately after 10 minutes the formation of red precipitate comes out. So a positive result, red copper oxide precipitate is formed, will indicate the presence of reducing sugar. So reagents, it comprises of copper 2 plus in slightly acidic medium. So here are the results for the Barfoed's test. As we can see, glucose and galactose yields a positive result since it is a reducing sugar. They are reducing monosaccharides. However, lactose should have been positive, but since it is a reducing sugar, but due to maybe it should have been heated more so that it would yield a positive result. Thus, it did not show on the on the experiment that was performed. We have the Benedict's test. So Benedict's test, 2 ml of the reagent were added with 5 drops of sample sugar solution, then it was placed in boiling water for 2 minutes, then was cooled. Benedict's test, it is a general test for reducing such sugars. So positive result is indicated by red precipitate. So reagents, copper 1 sulfate, sodium nitrate, sodium carbonate in mildly basic solution. Basically, that's the Benedict's reagent, an application of which is the diabetes urine test, Benedict's test on beer. So diabetes urine test is simple and effective method of ascertaining the presence or the amount of glucose in the urine. Benedict's test on beer. So since beer contains degradation products such as aldehydes and ketones, so yeah, it's tested on Benedict's. So these picture, as you can see, it contains numbers. So those colors, if it yields that certain colors in the experiment, it means it contains like such. For like in orange, we have 2.6 to 3.5% of sugar present. That's what it means. So here are the results from the experiment. We can see that glucose yields a positive result since glucose is a reducing carbohydrate, so it forms a red color, which means it has 3.6 to 4.0% of sugar. Then for galactose, it forms a green complex, positive, so it is a reducing monosaccharide, about 0.6 to 1.0%. Sucrose, it has no reaction since it is not reducing. Lactose forms a green complex since it is a reducing disaccharide and it contains about 0.6 to 1.0% of sugar. Starch, negative, since it's a polysaccharide and it is not a it's not a reducing sugar. So yeah, again, lactose is a reducing sugar because of the anomeric carbon of the glucose unit is free to interact with the oxidizing reagents. So that was the last test that were performed in the exercise 5. So now, let's proceed to the identification of the unknown. So in the identification of unknown, we have a schematic diagram that was used of qualitative tests for the identification of carbohydrates. So there were 6 sugars that were given for us, 2 for each group. And these sugars that was given were galactose, lactose, sucrose, glucose, and arabinose and fructose. So, with the use of the diagram, we identified what sugars of unknown we have. So, first, the schematic diagram. So, this diagram, as you can see in the picture, Molish test, it is a general test for carbohydrates, as what we have discussed. Iodine test, it is a test for polysaccharides. Blue color for starch, red for glycogen, and erythrodextrin. Barfoed's test, so it's a test for distinguishing reducing sugars or for both monosaccharide and disaccharide. So if no precipitation, then we could conclude that it is sucrose, and we could do a further test of which is the Benedict's test. Benedict's test is a test for reducing sugars. So sucrose is not a reducing sugars, hence no reaction. Next, if it forms a red precipitate in 5 to 7 minutes, it is a monosaccharide. Hence, proceed to bile-orsinol's test. It's a test for pentoses and hexoses. So if green solution, it is a pentose. Then proceed to a Selewanov's test to see if the sample is either arabinose or fructose, so faint red color for arabinose, red color fructose. If colorless or faint color, fermentation test. So if it has the formation of bubbles, it is a glucose. If not, it is a galactose. So if back again to Barfoed's test, if there is a formation of red precipitate in 7 to 12 minutes. It is a disaccharide. So fermentation test is further used. So if there is a bubbles formed, it is maltose. If none, it is lactose. So first, unknown A. So in the picture, that was the test that were performed. So Molish test, we did not really perform it, but it is expected to yield a positive result. And as for iodine test, we skipped that test since in the samples, in the unknowns that were given, we don't have a polysaccharide sample, so it would be just useless. So Barfoed's test, it yields a positive result. For bile orsinol's test, it yields a negative result. Sa Lewanov's test, it has a faint color, so fermentation was further performed and there were no bubbles. Hence, the sugar is known to be galactose. So next unknown is the unknown B. Molish test, positive, of course. Then Barfoed's test, there was red precipitate formation like within 11 minutes. So it is a reducing disaccharide. Thus, a fermentation test was further performed to see if it's either a lactose or maltose. So there were no bubbles, so hence the unknown B is a lactose. Next, for the unknown C, Molish test, positive. Barfoed's test, no reaction, so we proceed to Benedict's test. It has no reaction too, so the sugar is sucrose. But they also performed Seliwanov's test, and it turned into dark orange. This is because sucrose is a disaccharide and it contains fructose. Thus, the formation of that color. So, the conclusion, unknown C is a sucrose. So, next unknown is the unknown D. So, Molish test, positive. Barfoed's test, formation of red precipitates in 6 minutes. Bile arsenal tests were performed. Then, actually, this is negative test. It did not form dark green, so it's negative. Hence, it is a hexo sugar. Then fermentation test was performed. Thus, Seliwanov's test after the bile arsenal's test, it yield a faint yellow. Thus, it needs fermentation test. Then, it was found to be positive since bubbles were visible, or there were bubbles formation. Hence, the unknown D is a glucose. Unknown E. So next, Molish test, again positive. Barfoed's test, positive within 3 minutes. It is a reducing monosaccharide. Then bile arsenals, it is a positive test because it turned into bluish green. Then Seliwanov's test, it formed into faint pink complex. Hence, the sugar E or unknown E is arabinose. So lastly is the unknown F. So Molish test, positive. Then Barfoed's test, red precipitate formation with under 3 minutes. Hence, a reducing sugar monosaccharide. Yeah. So next, bile arsenal's test was performed. It was found to be negative. Hence, it is a hexose. So Seliwanov's test was further performed, and it was found to form a bright red complex. Hence, the unknown F is fructose. That's it for experiment 5 and 6. I hope you learned something from my report, and if you do have questions, don't hesitate to comment down below. Thank you for watching, guys. Thank you for watching. Block D. Block D. Block D. Chittin, 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 chittin.